So now, when it came time to do the 1 millivolt EPSP at the SOMA, that raises its own issues. Let's go ahead and if we saw this, let's go back to subthreshold, say 0.005 nanosiemens, and we look at that potential, we look at that depolarization there. It's about 6 or 7 millivolts. No, it's not. We're starting at minus 65 and we're going up to minus 56. That's about 9 millivolts depolarization. So if we cut that by, say, a factor of 10, and we can then look, but let me change the onset to 10 milliseconds like I wanted to do, like I should have done. And let's do that init and run. And let's do view equals plot. There we are. We have a nice stable membrane potential and what looks like a fairly stable membrane potential out in the apical dendrite. So here is our EPSP at the SOMA, and it's going from minus 65 to minus 64.25. So I need to make it about 1/3 larger. So I'll multiply that by 1.333, and we'll see what we get. Okay, view equals plot. Starting, we go from minus 65 approximately to, well, that's pretty good. So already we have a strong hint that the cell is quite linear. Because in order to, I started out with an EPSP that was about 30%, about 1/3 too small. And that was just based on a guess. I guessed that since I started out with a 10 millivolt EPSP, if I just cut that conductance by a factor of 10, I'd be close to a millivolt. Well, I was close, but I was still about 30% off. So then I thought maybe if I increase that conductance by 1/3, maybe I would get my 1 millivolt EPSP. And sure enough, for this signal that's on the order of about a millivolt peak amplitude, the cell is acting pretty linearly in the cell body. Okay. And now the natural question comes up was, well, is that just a peculiarity of the cell body? Is there anything about the rest of the cell? Can we say anything about how synaptic charge spreads into the rest of the cell? Can we draw any inferences about linearity of the model cell in this range, this low amplitude signal range? So the first question everybody asks when they say, I've got a synapse located at X point, and we know it's a this or that type of synapse activated by such and such a transmitter, everybody says, all right, well, how effective is it? If it's an excitatory synapse, how much does it affect the membrane potential at the soma? And how much of it affects other membrane elsewhere in the cell? So almost immediately, the experimentalist starts to wonder these things. And we can do these experiments and define and see these responses and measure these amplitudes in a model very easily. So we wanted to measure at the midpoint of apical 0 and of apical 2. We already have the midpoint of apical 2 here. We can add with plot what? The midpoint of apical 0. Apical 0. Okay, and I can move that text. Come on, move. Maybe it's not moving. It's not moving because I haven't selected move text. Moving. Okay, so that's apical 0. I'm going to arrange these in a kind of a topographical arrangement that soma, apical next to the soma, apical further away from the soma, and we also want to measure at the one end of the basilar, okay? the one end, the farthest end of the basal branch here. So let's do that with plot what? And I can just change that. I know its name is BAS, and I know the location is 1. So there we go. How come I didn't get it? Did I get it? I'm not seeing it. I'm not getting it added. Go back to crosshair. Now try plot what? Oh, there's no base 0. And I bet you dollars to donuts that neuron's interpreter window, I should say, I will bet you that the interpreter is complaining. Oh, it has been complaining. It says not the right number of subscripts. That's because there's only one branch called base. It's not an array. It doesn't have a subscripted name. So I just come back up here and get rid of the bracket. And now I click on accept, and no more complications. No complications. No more complaints from the interpreter. And I'll click on that, and we will now see. I'll get that back, and now I'll have to move something. Move this last one. There's base. There's apical. Okay, it'll be soma in the middle, base, apical 0, apical 2. And now let me do an init and run, and I'll do a view equals plot. And there's, but they're all the same color. I can change that to make things more interesting. I will change the base. I'll make that black. Where is base? Black. I'll make the apical 0 blue, which it is, and I'll make the apical 2 this kind of brownish, reddish, orangish color. Go back to crosshair. And at this point, I can make my measurements very nicely, and I'll know what I'm measuring from, okay? And so the exercise then becomes a matter of repeatedly showing the shape, moving your synapse to a new location, okay, like apical 0, 0.5, and now you know which trace is going to be bigger when we have our synapse attached to apical 0 in its midpoint. The trace at the apical 0 is going to be bigger because that's where we're dumping all of our current, and that's where the voltage change is going to be maximum. And if I do a run there it is. Sure enough, it's bigger. And I can do a view equals plot, and now I can come back and I can make my measurements, okay? And I can move it out to here, and sometimes I get it right, and sometimes I have to click a second time, and now the biggest one is going to be the one at apical 2, and that's really much bigger. And then finally I come out to base, and I can run that simulation and make those measurements there, okay? So I assume everybody did that or tried to do something like that. And how much of the EPSP spreads to these locations? 2 significant figures. Move it, but don't change its tau onset, etc. To a different location, and make this, repeat the measurements. Okay. And so, and record your data in a table. Okay. And that table is a good table to keep your data in. But when it comes to analysis, the first thing is, how do you make that information useful to you? And it's not a very convenient table to work with, but at least it's a complete record of your experiment, and you or your lab chief is going to want to see your notebook at some point, and they'll want to see that you pursued a clear experimental protocol and you made nice recordings. And if you're generating this with a program, you'll want to have your findings extracted somewhere and appear in nice organization as well. So let's do the following. Let's take a look at these questions, and let's take a look at the raw data. The first thing I think is easiest to do is to address this first question. Okay? Does an excitatory synapse act more like a voltage source or a current source? All right, so to do that, I'm going to take my experimental data and I'm going to look at a particular view of it. Actually, the first thing, yeah, I'm going to do data and analysis. Now, let's look at the experiments first. So these are the results of my different experiments, and this is for each of these figures is for a different pair of locations. So when I was when I had a synapse attached to the soma and I was recording the EPSPs at the soma and A equals 0, or switching that the synapse would be at A equals 0 and then recording at the soma. These are the results that I got, and you can see where the upstream electrode was, where the synapse was attached to, because that's the one that has the bigger trace, okay? So I'm recording membrane potential in the middle of the soma and membrane potential in the middle of apical 2. And when I had my synapse attached to the middle of the soma, the somatic trace, the somatic membrane potential was the one that was biggest, and the potential out in the apical is the one that is the smaller, okay? And then I swapped which site had the synapse and where I was recording. So now I made the synapse out in the middle of apical 0. Then the apical 0 had the bigger membrane potential response, and soma had the smaller response. So the color of the trace here is the location where I'm recording from, and whether the trace is, whether the potential at the soma is bigger or in apical 0 is bigger, tells you where the upstream electrode is, where the charge is being dumped into the cell. So you can see the smaller responses then are always the downstream potentials. So the blue trace is the downstream potential when I'm dumping charge in the soma. The red trace is the downstream potential when I'm dumping charge into apical 0, in the middle of apical 0. And you'll see that regardless of where's the upstream and what the downstream is, as long as, if I look at the other location, they're pretty much the same in waveform. So between the soma and apical 0, synaptic current entering the soma produces a response at apical 0. Taking the same synapse and attaching it to apical 0 and seeing what happens at the downstream site, I'm getting the same effect at the downstream site, whether the downstream site is the soma or apical 0. So this means that if a synapse in the dendrite can produce a 0.5 millivolt depolarization in the soma, if you could pluck that synapse away from the dendrite and attach it to the soma, same synapse, then activate it, it would elicit a 0.5 millivolt out in the dendrite at the location you were recording, at the previous location where the synapse had been, okay? So that kind of looks like reciprocity. okay? That looks like, it doesn't matter where in the system you attach the current source, you're seeing the same result at the downstream point. So if you had 2 points of interest, A and B, and you attach the current source at A, you get a result at B. Now, if you take the current source and attach it to B, and while you're observing at A, you get a very similar response at A that you had previously seen at B. Likewise, between soma and apical 2. When the synapse is attached to the soma, makes a big depolarization in the soma, but this little orange trace down here is the response in apical 2, and it's very small. And we measure it relative to its baseline here. We see we have a very small depolarization at apical 2. Now, if we take our synapse away from the soma, attach it to the apical 2 location, we get a big depolarization at apical 2 and a small response at the soma. And if we measured the amplitude of the response at the soma, which is now the downstream electrode, we see that this peak amplitude of this little response is pretty much the same as the peak amplitude of this little response. So that's looking like reciprocity between the soma and apical 2. And if we do the same experiment between the soma and the distal end of the basilar, if the synapse is attached to the soma, we get our 1 millivolt depolarization at the soma, but we get a smaller response at the distal end of the basilar. Well, we expect that because the distal end of the basilar is, it's farther away. We expect some attenuation in the delay. Now, if we pick up our synapse from the soma and transplant it out onto the distal end of the basilar, we get a big depolarization in the basilar. No surprise. We expect that. The basilar is a narrower branch. We expect a higher input resistance. We expect a big depolarization out there. And we do see a smaller response at the soma. You can see there's a little bit of red there, okay? But it's practically the same amplitude now in the soma that had been in the distal end of the basilar when the synapse was at the soma. So we're seeing really good reciprocity between the soma and the basilar location. And likewise, if we take the other pairs that are possible, how reciprocal are signal interactions between apical 0 and apical 2? Pretty darn reciprocal. These are the smaller responses. These are the downstream responses, okay? And the downstream response is the same regardless of which of these two locations is upstream. Very, very close, very similar. Likewise, between apical 0 and basilar, these are the downstream responses. They're small in amplitude, but they're very similar in time course and amplitude, regardless of which of these is the upstream location. And again, between apical 2 and basilar, we see that the downstream responses have very similar amplitudes. So we're looking at what looks like reciprocity. Okay. Now, what is reciprocity? Well, it's a property of linear systems. Okay. And it was first described in the 19th century by physicists. And it's a hallmark of a linear system. So we have a suspicion that there's reciprocity, that this cell is quite linear for low amplitude signals. Let's take a look at the data. Now that I've planted that idea in your head, and we'll come back to it. Okay. So I don't need the side pane. Side panel's not needed. So these are the raw data from the experiments, and they're rearranged in a way I think is going to make more sense than, in retrospect now, than the way I thought. And the way I thought was pretty much the naive approach. You take one stimulus site and you record at all the recording sites and you record all those recordings. Then you take a different stimulus site and you record all the recording sites' amplitudes and you throw those into a table. But this now I think makes a bit more sense, a matrix type of arrangement where we show the rows correspond to synaptic locations and the columns correspond to measurement locations. And if you look here, you look along the main diagonal And you see the biggest responses are always when the injection site is the same as the measurement site, which is not a surprise. Okay? You're dumping the charge at the same point you're measuring the resulting potential change. You expect that to be the case. Okay? You'll also know I threw in one additional column here, which was the peak current. So these are all the peak amplitudes of the EPSPs that are observed at the different locations, and these are the peak currents. And you'll see that the peak current does change a bit when you change the synaptic location. So when the synapse is attached to the soma, it delivers more current than when the synapse is attached to the basilar or the distal apical. Okay? Now, why would that be? We'll talk about that later, maybe. But what was our first question that we wanted to answer? And that is, does an excitatory synapse act more like a voltage source or a current source? Okay. Well, if it acts like a voltage source, then the local voltage that it produces at the location where the synapse is will be relatively insensitive to synaptic location. So if it produced a millivolt here, it should produce a millivolt here. If you attach the synapse here, and if you attach the synapse here, it should produce a millivolt here out in the distal apical. And if you attach it to the distal end of basilar, it should produce a millivolt there. And we can look at this table and we can see that's clearly not the case. But let me just go ahead and gather the local depolarization, the depolarization at the point of synaptic attachment, and the peak synaptic current delivered by the synapse as a function of where the synapse is located, okay? So if the synapse is located in the soma, you get a millivolt depolarization. If it's located at the apical 0, you get almost 2 millivolts out in the middle of the apical dendrite, 0. If it's at the middle of the distal apical dendrite, you get about almost 5.6 millivolts. If it's at the distal end of basilar, you get 1.7. So clearly it's not acting like a voltage source because it produces a different depolarization depending on where it's located. If we look at the currents that it's delivering pretty close to the same current though. That's not much variation. It's a significant variation, and there's a reason why it happens. Okay? Does anybody know why it happens? Why does this variation of peak synaptic current occur? We're taking the exact same synapse and moving it to different points in a cell, an experiment that you can't do in the wet lab, but you can do in a computational model. Why is the peak synaptic current changing as a function of synaptic location? Anybody have an idea?
Depending on the input resistance where you are injecting.
Yeah, you're very, very quiet, very faint. Can you speak a little louder?
Depending on the input resistance at the location where we're injecting.
Absolutely. Are you an experimentalist, by the way?
No.
You're a physicist, engineer?
My PhD was in computational neuroscience, just modeling.
Well, okay. All right. Of course. Of course. I hope other people also had that same intuition, and maybe we're just shy. If we, when we do the summer course, and we have people actually physically present, we go around the circle and everybody gets a chance to answer a question. Everybody gets a chance to propose hypotheses. Sometimes we have things where people come up to the board and sketch things. We actually get different opinions as to what's going to happen during a computational experiment and make little bets as to what the outcome is going to be. It gets to be more fun. It's hard to do that remotely, but I'm glad that there are some people who will volunteer and speak. Don't be shy. Okay, so yeah, the input impedance is a bit different. And because the input impedance is different, but the input impedance is vastly different. Okay, it's huge. It looks to me like for the same injected current, we're getting a depolarization almost 6 times bigger. And actually, it's not even the same injected current, it's a bit less. So the input impedance out here in apical 2 looks like it's almost 6 times bigger, but the current reduction is only less than 10%. Why is that? Why is the peak current only less than 10% reduced? Okay. Anybody have an idea? Nope. Okay. Well, let me go back and do the experiment here. And what I'm going to do is current. And what I'm going to plot is, of course, the alpha synapses. Oh, it doesn't have one. Ta-da! Cancel. So we need to turn this into an alpha synapse. And its onset is going to be at 10. The time constant is 1. And Gmax, we're going to say is 0.00 what?
6.
0.006 approximately. Anybody have a better value? That's close enough, I guess. Hit and run. And that was for our 5. Gave us a subthreshold and divide by 8. Whoops. 5 divided by 8 was close to a millivolt. And very close, yeah. 6 is
Around
close enough.
0.067 microsiemens for the 1 millivolt EPSP.
0.067, okay? And that gives us a millivolt. Good, okay. So there is our EPSP. And what is the driving force for synaptic current? Let me see, let's plot the current. Alpha synapse, 0, I. And let me move that. And let's do a run and do a view equals plot. Okay, there's our, move this again so it's not in the way. There's our synaptic current. What is the driving force for synaptic current? The driving force for synaptic current is the difference between the reverse potential of the synapse and local membrane potential. Okay? So the equivalent circuit, I don't know. Do I want to annotate? Do I want to draw? Gee, I don't want to get involved in drawing on this darn thing. But the equivalent circuit is basically a battery in series with a variable resistor. Okay? That's what a synapse looks like. It looks like a battery with a reversal potential. In this case, it's a 0 millivolt battery. One end is attached to ground. The other end is attached to a variable conductance or variable resistance, which is then attached to the inside of the cell. Okay, so the driving force is the difference between this and minus 65 millivolts. Well, what happens to the driving force during the EPSP? Let's plot the driving force as a function of potential, and we would see, well, I'm not going to plot it, but it would go down from 65 millivolt to 64 at the peak of this EPSP, right? That's when the synapse is at the soma. Now, if we move the synapse out here, that's a little fussy. There we are, apical 0.5. Hit and run, let's plot that voltage. Our cell is a B cell, it's B cell 0, it's apical 2, and it's V, not at that location, but at 0.5. And now let's do that. And it's big. And our peak depolarization is, wow. Okay. So it 65 minus 59.4. So it reduces by 5.6 millivolts. Okay. So the driving force starts out at 65 millivolts and drops down to, to 59.4 millivolts. And that's a significant change. That's a 10% reduction of driving force. So the current through the resistor is being driven by less and less of a driving force. We should get less and less of a current. And if we approximated this as a triangle and then said, let's take half of that, find out what the average driving force is, okay, we could get a reasonable approximation to the average, to the charge that actually, the average current that flows, the charge that actually enters the cell, and we could probably predict fairly closely the peak depolarization. Okay, I'll leave that as an exercise to computational neuroscientists, mathematicians, physicists, and engineers. Okay, but this is why we get a reduction, but it's only a small reduction. We're getting a roughly 10% reduction of driving force, actually slightly less than 10% reduction of driving force. What is our reduction of current? Slightly less than 10%. Okay, the peak synaptic current is going to occur pretty close to the peak of the synaptic conductance. And the driving force at that point we could calculate, and we'd find out we're getting, that would account for most of this drop. That would account for this drop. Okay. All right, so even though the input impedance is huge, we're only getting a relatively small reduction of driving force, and but that's enough to reduce the synaptic current. Okay, so excitatory synapses act more like current sources than like voltage sources, and that if you go back and you look at all the classical papers about electrotonus, they're all obsessing about, well, if you have a 1 millivolt depolarization here, how many millivolts depolarization is it after it spreads to over there? Their real question, maybe they should be focusing on synaptic current. Maybe they should be saying, if we have a certain amount of current entering here, what's the depolarization as a function of location? Okay. In other words, instead of thinking about voltage attenuation ratios, they maybe should be thinking about transfer resistances, transfer impedances. Okay, what's a transfer resistance? What's a transfer impedance? Well, we know what an input resistance is. If you're at a particular point in a cell attached to it, you inject a current, you see a particular change of membrane potential, divide the delta V by the delta I, that's the input resistance at that location. Now let's say you're injecting current at one point A, and you're measuring voltage at point B. So the transfer resistance from A to B is the current you're injecting into A divided into the voltage you see at B. That's the transfer resistance from A to B. Now, if you swapped the injectant and recording sites, you could measure and calculate the transfer resistance from B to A. You inject a current at B, you see what the voltage change is at A. The transfer resistance from B to A is the voltage change at A divided by the current injected at B. If the system is linear, the transfer resistances will be independent of the direction of signal flow. Okay, so it doesn't matter which one is the upstream electrode, the transfer resistance should be the same. Well, is it the same? Okay, let's take a look. When we look at our data table, This is the same data table we had right up here, only I've now colored the cells. If we look at the table, we see an interesting pattern. For any pair of locations, the observed EPSP, these are the locations. The observed EPSP for any given pair of locations is identical or nearly identical whether the synapse is at A and the EPSP is recorded at B or vice versa. So if we have the synapse at the soma and we're recording at A equals 0.5, or we have the synapse at A equals 0.5 and we're recording at the soma, we see these two voltages are nearly identical. In fact, they are to two places. Now, if we had this, again, the same for soma and apical 2. That apical 2 here is downstream. For this cell, the soma is downstream. Okay, we see pretty close to the same. There is a difference, but it's pretty close to the same. If we compare what happens when apical 0 is upstream and apical 2 is downstream versus what happens when apical 2 is upstream and apical 0 is downstream, we see pretty much that's pretty close. And likewise between soma and the distal end of the basilar. If the soma is upstream, we get 0.67. If the soma is downstream, we get 0.66. Those are very close to being the same. And likewise for basilar versus apical 0, 0.3 millivolts. Or between basilar and apical 2, 0.4. Not exactly the same here, but very similar. Did you expect that? Who didn't expect that? Show of hands. Who wouldn't have expected that? The rest of you have Guillain-Barré syndrome, huh? Brachial palsy. No? Well, okay, we have 3 people who are alert and honest. Okay, you get well. All right. The implication here is still, again, is suggesting electrical reciprocity. It doesn't prove reciprocity. Why? Because we're not actually injecting the same synaptic currents. Because we know that synaptic current depends on location. So this difference when apical 2 is upstream versus when soma is upstream might be due to the fact that when apical 2 is upstream, less charge is getting into the cell. So if less charge gets into the cell because there's less driving force, then the soma EPSP is going to be smaller than when the soma is upstream and apical 2 is downstream. Okay? So maybe we should be actually calculating the transfer impedances, not just looking at the EPSP amplitudes and comparing those. Let's actually calculate the transfer impedance. So the transfer impedance from soma to soma that would be 1 over that, which is about 20 what? About 22, 23 megohms. Okay? So that's actually the input impedance of the soma. Let's look at our next table here. Oh yeah, look at that, 23.20. That's the input impedance of the soma. That's the input impedance at apical 0. That's the input impedance at apical 2. And sure enough, apical 2's input impedance is about 5 times what it is at the soma. Okay, maybe closer to 6. And the input impedance at the distal end of basilar is pretty significant too. The transfer impedance from soma to apical 0 is the depolarization at apical 0 divided by the peak depolarization there, divided by the peak amplitude of that synaptic current that entered the soma, and it's 10.44 megohms. The transfer impedance from apical 0 back to the soma is the peak depolarization in the soma divided by the peak current, peak synaptic current that entered at apical 0, it's 10.59. And if we, and that's within 1%. Well, that's within 1%. No, 1%? Yeah, 1.5%. That's within 1.5% of being perfectly linear. Likewise, we find, oh, that is exact between apical 2 and soma. Not even 1% off. Those transfer resistances, transfer impedances are identical. And again, remember, I'm measuring peak amplitude of a complicated waveform. So it's an impedance, not just a resistance, okay? Between the basilar and the soma, it's within, about 1%. Between the distal end of the basilar and the apical and mid of apical 2, apical 0 also. And likewise between the end of the basilar and apical 2. The transfer impedance is independent of direction of current flow within about 3%, okay? So for small signals like individual EPSPs, the subthreshold behavior of this cell is nearly linear, even though we know it has active currents. We know it's got Hodgkin-Huxley in the axon and the soma and in the apical dendritic field. And the implication of this is that small signal analysis can be used to understand how it is that subthreshold signals propagate in a neuron. And I will just leave you with that idea now to think about. We will return to it in the future because Neuron has a software and GUI tool that you can use to do small signal analysis of cells. And it can take into account the active gating properties of the cell, okay? And it allows you to make measurements of impedance, transfer impedance, impedance over a path, all sorts of interesting things, voltage attenuations, charge transfer ratios, voltage transfer ratios, all very straightforwardly and easily, and taking the gating of active currents into account.